Hello class, today we are going to be learning about how neurons communicate with each other. So you will need a blank sheet of paper and something to write with and perhaps some colors. At the end of this, you'll take a picture of your completed notes and put it into your Health Google Drive. So please label this neuro, which means neuron. So this is going to be neurotransmission, and that is going to be communicating between two neurons. So let's go ahead and sketch what that looks like big picture, then we're going to zoom in on a more molecular level. So let's go ahead and draw two neurons, like that. I'm going to put in this, like that, right, and then all right, please identify the following features. A, the dot in the middle. B, the long part in the middle. C, the surrounding substance. D, and this location. E. All right, so this right here is going to be called the presynaptic neuron. And what we're going to do is we're going to sketch next to it the postsynaptic neuron. And remember, they can never touch each other because they have electricity to them. Let's go ahead and go here, sketch. Remember, each neuron can make up to 10,000 connections with other neurons. All right, we're sketching in now the axon terminals. There are many axons, many dendrites, but the basics are dendrites, nucleus, axon terminals, and if you're not a hardcore drug user, you've got a really nice and healthy myelin sheath that keeps your electricity on your neurons. So this is going to be called your postsynaptic neuron. And the gap in between neurons is called the synapse. Sometimes you'll hear synaptic cleft. Okay, so that space right there is significant. This is going to be called your synapse, which is just your space between neurons. All right, what I want to do is show you a better image of what I sketched. So here you have a presynaptic neuron, postsynaptic neuron. This space right there is the synapse. That's a pretty good image right there as well. Remember, this is a cell, so it's going to have a mitochondria, this whole light yellow thing, that's all cell membrane. All right, and this is what I was hoping for with the image that we just looked at. So what we're going to do right here is zoom in on one of those synapses. Okay, so I'm going to put surrounding. I'm going to go down here. We're going to label this a synapse. All right, so it can look somewhat like this. We can have a big bulb-shaped axon terminal, and then we can have a receptor dendrite like so. So please go ahead and label which type of neuron each of these are. So this will be neuron number 1. Go ahead and label what that is, and neuron number 2. Please label what type of neuron that is using these terms up here. This right here is your presynaptic neuron. We're just taking a zoomed in look at it. And this over here is after the synapse, so it's the postsynaptic neuron. All right, let's go ahead and be true to our picture. Let's go ahead and cover this axon in myelin. And we'll do the same right here. It's not a full picture of it, but that's okay. That is not the purpose. So this right here will be the axon. This right there is our myelin. Same over there. That's our myelin. Okay. This right here, this general region, is our axon terminal. We have our synapse right there. This right here will be one of many dendrites. The electrical signal that's going to be propagated is going to be moving in the left to right direction. So this is going to be the direction of signal travel. All right, so what I want to do right now is focus on our presynaptic neurons. So I'm going to create a key over here. A circle like this is going to equal a vesicle. And I'm going to make orange dots like this, just a general neurotransmitter. So I'm going to label that NT, where NT stands for neurotransmitter. So I'm going to create these vesicles inside of my axon terminals. And inside of these vesicles, I'm going to place loads of neurotransmitter. Remember, these were created from the cell's nucleus. It sent out the code to make this neurotransmitter. It could be dopamine or serotonin. Those are the most popular ones. And this is where bio and health meet. This vesicle was made by the Golgi apparatus, and it is just basically cell membrane, a circle of cell membrane with neurotransmitter inside. So let's go to this image here. Cell vesicle looks like that. You guys can see the green phospholipid heads, the yellow tails. Here's a picture of the vesicle. The pink would be the neurotransmitter inside. Okay, now let's focus over here on the dendrite side. The dendrites over here are going to have these little receptor cups on them. And just like in biology and enzymes and things we talked about, these receptor cups are perfectly suited, like a puzzle piece, to attach the neurotransmitter that will be associated with it. So this right here is called a receptor. And these are found on dendrites. This is going to be where your NT, your neurotransmitter, attaches. Okay, always, I'm just going to define it over here because sometimes when you use an acronym, you always have to define it at least once on the paper that you're on. So this is going to be your neurotransmitter. Okay, so now we have the stage set. We have our neurotransmitter, we have our receptors. Now we need to somehow pass the information from this neuron over to this neuron so we can continue the signal of whatever that person is feeling at the time. If this were dopamine, you'd have excessive pleasure being felt. The more dopamine in the synapse, the more pleasure the person would feel. This could be serotonin. This could be giving a person an excited feeling about life. The more serotonin, the more that feeling becomes. Okay, you guys, I want you to get a new piece of paper out, but if you have space on your old paper, you probably need about 10 lines at minimum down here. I need more space on this paper, so I'm opening up a new paper. I will also put this one on my Google Drive, and I'll grade it from there. So this is going to be neurotransmission. And this is going to be step by step. So over here, I'm going to have a picture. Over here, I'm going to describe what is happening. So I don't have a ruler right now, so I'm going to let my obsessions go. Just draw a line down here. This will be images. Over here will be steps. All right, let's begin by focusing the document. All right, step one. So I'm going to go draw over here. That is my axon terminal. I don't know why I put that line there. Here is my dendrite of my following neuron. So this one is going to be the neuron that is on. This is going to be the neuron that is off. So we're going to define a few acronyms before we continue. AP is going to equal action potential. And basically, this is the same as biological electricity. You have a lot of electricity in your body. You have electricity in your brain. You have electricity in your heart. These are the things that keep us moving and our heart pumping, etc. That's why when they try to revive someone, they shock their heart because it's electrical. Your brain is electrical as well. And then obviously we're going to have our NT is going to be neurotransmitter. Then we're going to have our pre-S is going to be our presynaptic neuron. And our post S is going to be our postsynaptic neuron. So hopefully with these abbreviations, we'll be able to move through these steps, and there are not very many, very efficiently. So let's begin. Step 1. In order for this to be on, the action potential has to be on the neuron. That's what defines on. So I'm going to make a little lightning bolt right here. And that lightning bolt, I'll put that right here as well. That's going to represent action potential. So step 1 is that you are going to have an action potential on the presynaptic neuron. All right, somehow we need to get the action potential from over here to over here. So we need to now focus on the vesicles. Let's go ahead and sketch this picture right here. We can make them large, there's not too many steps. Okay, the action potential is here, but it continues to propagate through here. So when it gets to the axon terminal, it's going to cause these vesicles right here to get excited. Okay. I'm going to draw my vesicles. They have my neurotransmitter in them. The action potential, I'm going to use the same color, is going to be transferred to the vesicles. So I'm going to surround the vesicles now with electricity. That's going to cause them to get excited. So what's going to happen is these vesicles are going to do a function called exocytosis, which we talked about a few months ago in bio class. Let me show you what that looks like. Here's exocytosis right there. Okay, here's my vesicle. Here in the green are my neurotransmitters. They are going to get excited. There we go, watch. Exocytosis, and boom, neurotransmitters are in the synapse. Then they get reabsorbed, they get excited again, and out they go into the synapse. One more time. That is my brown vesicle made of cell membrane. The action potential turns it on. Boom, in the synapse. So next, the action potential is passed on to the neurotransmitter vesicles. Okay, it's all about passing. It's like hot potato. The action potential is a hot potato. It went to the axon terminal. It went to the synaptic vesicle. Now that's going to cause exocytosis of the synaptic vesicles. So I'm going to put, this is step 3, AP on synaptic vesicles causes exo means exit, cyto means cell, so it's going to exit the brain cell, causing exocytosis. Okay, step 4, neurotransmitter is now in the synapse. All right, so I'm going to put my action potential, that's not my action potential, I'm going to put my neurotransmitter in the synapse. So this neuron is still on, this one is still off, okay? So the action potential is about to be passed over now to our postsynaptic neuron. So now we're going to focus on the post side. So go ahead and draw an empty pre side. We're going to focus now on the post side. So on your postsynaptic neuron, I want you to attach these receptors, okay? These are receptor cups. And I want you to take your same neurotransmitter color and attach your neurotransmitter to the receptors on right here, the dendrite. Okay, this is a molecular on switch. The minute those attach, you now have passed successfully the signal from point A to point B. This is on. This one no longer has action potential on it, so it is considered off. So now you have successfully turned on your neighboring neuron without actually ever touching it physically neuron to neuron. They just sent this chemical on switch called the neurotransmitter. So neurotransmitter is now in the synapse. Number 5, neurotransmitters attach to receptors on dendrites on the postsynaptic neuron. Only a couple more steps. Now the postsynaptic neuron is considered on. The signal continues to travel. And this signal can be you hearing me right now, or you seeing, etc. There's so many signals happening in your brain at one time. And remember, every neuron can have about 10,000 connections, and you have about 100 million of these in your body. So we're talking a very small scale, what we're looking at right now. Okay, so it has successfully turned on the next neuron. Now we need to deal with what to do with the neurotransmitter. So step 8 and the final step, the used neurotransmitter has 3 options. And I will sketch those right here. I'm going to add a new element right here to the axon terminal. This is called a transport protein. When someone's using cocaine, the cocaine comes in and blocks the transport protein, so you keep the happy serotonin signal between the synapses, so the person gets this overload of serotonin because they can no longer transport the neurotransmitter back into the presynaptic neuron via transporter protein. Okay. So it has 3 options. The first one is the neurotransmitters return to your presynaptic neuron, where it's pre-S period neuron, using a transport protein. Antidepressants do something similar without the horrible side effects, is that they keep the reuptake channel, which is also another name from this. They block it, so you keep that serotonin in your synapse, so the person has that excitement about life. The second one is short, it can dissolve. Or lastly, final point, it can return via endocytosis, which is the exact opposite of exocytosis. So I'm going to pause the video. Actually, I'll stop the video. You can get caught up on the notes. Take a picture, please, and put them on Haiku. Not on Haiku, I'm sorry. Upload it to your Google Drive, and I can see everyone's pictures just from one single website. Have a great day.